Good morning, all. Myself, Mrs. Priya Sonawani, and I'm going to present a paper in today's International Conference on Sustainable Development Goals, Key Issues, Opportunities and Challenges in Environment, Agriculture, Population, Resources, Economy, and Social Sciences. So I am very happy to present my paper over here. The title of my paper is Biological Treatment for Synthetic Pyrethroids, that is lambda cyhalothrin degradation in the farmland soil. Uh, as all of us know that the major environmental concern, it is the contamination of various types of ecosystems due to pesticides, and synthetic pyrethroids are one of the type of the insecticides which can control the insect pests and disease vectors since long time. These synthetic pyrethroids are useful for controlling the agricultural pest as well as the mosquitoes, hence it is widely used. The use of these synthetic pyrethroids in India, it has sharply increased since many organophosphate products such as malathion and parathion, they are being phased out of the agricultural use. So with this extensive application, many adverse effects are also observed, and that's why we need to focus on the study that these synthetic pyrethroids must be removed, they must be eliminated from the environment, or at least they must be at the safe level. Along with this, we know that these synthetic pyrethroids, they are safe for humans, but exposure to high concentration, it may cause the human health problems. In addition to this, the bees, fishes, then crabs, tadpoles, or other animals, they are also affected due to toxic effects. Therefore, it is necessary to eliminate and reduce the concentration of synthetic pyrethroids in the environment. In the present study, I am using the biodegradation. So here the target pesticide will be used as a sole carbon source and energy for microorganisms. Then to assess the microbial degradation of pesticides, the bacterial culture is isolated and it is characterized for the lambda cyhalothrin degradation potential in the contaminated soil. So we'll start with this. As we have seen that its use is widespread, hence it is responsible for soil and water pollution. Then various aquatic ecosystems are also polluted. These synthetic pyrethroid insecticides, they were introduced into widespread use, and it is for the insect for controlling the insect pest and disease vectors. So extensive application is there, and many adverse effects are also there, such as pest resistance, then residues in food and environmental contamination. The effects on health, they are also over there, like bioaccumulation toxicity is there, then immune suppression or endocrine disruption, then it may modify electrical activity in various parts of the nervous system, the neurotoxicity, lymph node and splenic damage, and carcinogenesis. So it is very necessary to develop the rapid and efficient disposal process to eliminate or minimize the concentration of synthetic pyrethroids in the environment. Various physical and chemical methods are also available to treat the soil contaminated with such hazardous material, but biological treatment, which is the most promising treatment, that is biodegradation, here especially the microbial degradation has proven to be suitable method for insecticide elimination. The materials which I have used, it is all of the analytical grade. The lambda cyhalothrin, it is trade name, it is popularly known as the the um, karate pesticide. It is dissolved in acetone solution and it was added to desirable concentration. Various concentrations I have used in the medium as a sole source of carbon. Then MSM medium, that is mineral salts medium, is used. It was prepared and then it was sterilized by autoclaving. Enrichment, isolation and screening of bacteria was done. For this, the soil sample was collected from the farm near Dindori Taluka, district Nasik in Maharashtra, and enrichment was performed by placing 10 grams of soil in 250 ml of Erlenmeyer flask containing 100 ml of sterile MSM media with the initial concentration of lambda cyhalothrin at 20 mg per liter and incubated in shaker incubator at 28 degrees Celsius for 100 rpm. After 10 days, the media turned red brown and 5 ml of aliquots were collected, and then it was transferred to 100 ml of fresh MSM medium, and here the concentration was adjusted up to 50 milligram per liter. Now again 10 days it was incubated, and later on the bacteria was isolated on the solid media plates. The extraction of lambda cyhalothrin residue for residue analysis was done. For this extraction and quantification, the media it was modified slightly by the from the method which was described previously, but here at different time intervals the triplicate populations were sampled for lambda cyhalothrin concentration analysis. The lambda cyhalothrin was extracted 3 times from the media with 100 ml of hexane. The hexane extracts from the same sample were combined, dried, and anhydrous sodium sulfate and concentrated by exposure to nitrogen gas near the dryness to on rotary evaporator. Then it was dissolved in 5 ml of hexane for GC detection. Detection of the metabolites was also done, and these were isolated from the culture filtrates of the organism grown in lambda cyhalothrin. The concentration was adjusted up to 100 mg per liter by extraction with acetonitrile. Before and after acidification to pH 2, that is acidic pH, with 2 molar HCl, and residue obtained was dissolved in hexane, and then it was identified and analyzed using GCMS system. Here after repeated enrichment and purification processes, we obtained approximately 7 strains of of the organisms with different colony morphologies from the farm soil samples. The isolated bacteria was gram negative, anaerobic bacteria, and the morphology of the colonies was also studied. The effect of initial concentration on lambda cyhalothrin degradation was studied, and it was shown that the lambda cyhalothrin degradation rate was found to be very excellent, and it was near about 90.4% at the concentration of 100 milligram per liter, and various degradation rates were also detected at different concentrations. So this was the degradation rate of lambda cyhalothrin. Then the metabolites after degradation were also identified with the GCMS system, and for this we have used the proper GCMS system using the Agilent MSD ChemStation software, and the compounds which were identified where there were 4 products identified after this. It was cyclopropane methanol, then 5-methoxy 2 nitrobenzoic acid, then 3,5-dimethoxybenzamide, and 5-amino-isothalic acid, and they have the different retention times. So here we can conclude that the bacteria which was isolated from the soil sample, it was collected from farmland on pesticide contamination, and it can utilize the lambda cyhalothrin as a sole source of carbon, nitrogen, and energy. So degradation rate reached up to 90.4% within 7 days under optimum conditions, and 4 metabolic compounds were detected, and this can give us the hint that there are complex redox reactions. They were involved in the lambda cyhalothrin degradation process. These were the references I have used for my study. And here I can end my presentation. Thank you so much.